I'm going to present the steps for a variant query in SNPnexus COVID starting from a single input. This input was obtained from the COVID-19 Host Genetics Initiative and represents variants identified from a meta-analysis between COVID-19 positive patients with severe respiratory complications and a population that was not hospitalized. Each query requires contact details to be provided for users to receive a notification once the results are available from the uploaded dataset. As you can see, SNPnexus COVID currently accepts inputs aligned to the HG38 genome assembly. Input variants, which can be uploaded as single nucleotide variants, or insertions or deletions, can be provided in several compatible formats. These different formats are presented in more detail in the online user guide. While in this example, the input will be variants identified from a meta-analysis, SNPnexus COVID also allows for variant data from multiple samples and files to be uploaded. An example of a multi-sample query is available from the next video in the user guide. Information pertaining to the genomic location and the reference and variant allele are pasted into the text area. Researchers can filter the uploaded variants by human genes with potential links to COVID from ONCODE and the Genomics England gene panels. In this instance, the analysis is run on all the variants uploaded without the application of a filter. A range of annotations can be selected based on researcher requirements. These comprise gene or protein consequences, resources geared at biological and clinical interpretation, such as GTEx, the human phenotype ontology, protein-drug interaction networks, and reactome pathways. In addition, annotation categories pertaining to effects on protein function, population frequency data, regulatory elements, conservation, non-coding scoring, and structural variations can also be selected. To maximize the results returned for this query, all the annotation categories are selected. The query is then submitted. Depending on the size of the upload, users can either stay on the upload page to access the results or wait to receive a notification email with links to the results. The first section of the results page provides researchers with a summary of their input and a list of filtering criteria available. On the left, the name of the dataset, severe versus population, the human assembly, HG38, and the number of variations mapped, 1,237, are presented. Full results can be downloaded in either VCF or text format to be viewed locally. Alternatively, researchers can explore the results using the interactive tables and plots provided. The filters available allow users to restrict their query based on defined characteristics, such as the type of variant, trait association, genomic consequence, GTEx, phenotype and disease association, so human phenotype ontology and ClinVar, drug, pathway, predicted effect, conserved regions, or genes of interest. There is also a section dedicated to all the graphical outputs generated. Plots available include plots presenting the proportion of input variants bearing different predicted functional consequences. For these results, most of the variants are identified as coding, with the majority classified as intronic. These can be viewed in more depth in the karyotype plots, where genomic consequence of the input variants across each chromosome is shown. There is also a bar plot presenting a summarized view of whether protein function is affected by amino acid substitutions, where variants are classified as deleterious or tolerated, that is to say, benign. These results can also be viewed as a karyotype plot for additional granularity. Furthermore, a summarized view of variant features is available. The information presented includes variant classification and type, the proportion of transitions and transversions in the data, classification summary, and the top mutated genes for the single patient upload. If multiple patients are uploaded as input, then this summary will present a cohort view of all the results. The Manhattan plot presents significant SNPs across the chromosomes. Again, a cohort plot is generated in instances in which a user uploads multiple patient files as input. Genes associated with variants present in the uploaded data are associated to biological pathways in the reactome pathway database, with pathways presented in an interactive Voronoi diagram. Here, enriched signaling pathways are highlighted in yellow. In agreement with the literature, we observe aberrant signaling by GPCR. Finally, SNPnexus COVID provides users with the opportunity to assess the pharmacogenetic potential of genes identified within individual patients or larger cohorts. The network diagrams generated present the protein of interest along with interacting proteins and potential drugs. Focusing on TYK2 and OAS2, we see that TYK2 is a potential drug target for fostamatinib. Fostamatinib, a spleen tyrosine kinase inhibitor that reduces immune-mediated destruction of platelets, is currently being assessed for safety for the treatment of hospitalized COVID-19 patients. Similarly, tofacitinib is also in phase 2 trials to assess its suitability to treat hospitalized participants with COVID-19 pneumonia. Moreover, SNPnexus COVID also provides the opportunity to view associated proteins and their potential as drug targets. For instance, drugs linked to JAK2 and HNRPK. Looking at heterogeneous nuclear ribonucleoprotein K, we can see that it's potentially druggable by the anti-malarial agent artenomol. OAS2 does not appear as a drug target, but we can see that this protein interacts with CLK3, which is the target for fostamatinib. All of the information used to generate the plots and much more is available to query from the interactive tables provided below. These provide information for all the annotation categories selected at the start of the query, so researchers can view overlapping genes, gene or protein consequences, results from GTEx, where multi-tissue EQTL plots can be viewed, these display multi-tissue posterior probabilities from meta-analyses against single-tissue association results. Phenotype and disease association, protein interactions, highlighting the target protein and target name which could be a potential repurposing candidate for COVID-19, pathways linked to the affected genes, effects of variants on protein function, the frequency of variants in normal populations, regulatory elements, conservation, non-coding scoring, and structural variations.